All right. Get on. Let's get this review done. No. Come on. Let's get this review done. Bobo? No. Sheesh. Mm-hmm. Da, 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 da. Da, 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 da. Na 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 na. I'm so sleepy. Oh yes, I'm so sleepy. Seriously, you should see my freaking dog. He's like going psycho. So like, so excited right now. I don't get it. All right, let's go. Okay. Yay, we've got a question. Yay, we've got questions. Questions. Hey, I could listen to you sing. Yeah. All right, let's get this bread using fermentation services. What? Let's get this bread using anaerobic fermentation centers. I don't know what's going on, Patrick. I just don't know. Okay. Let's get this bread using anaerobic. All right, Alicia, yay, we've actually got a question. Yay. Let's look at a question. Okay, can we go over oxidative phosphorylation? Yes. So first, let me just kind of write out the types of phosphorylation that exist that we've talked about. Phosphorylation, if you're talking ADP plus a PI, inorganic phosphate, being converted to ATP. That is just good old-fashioned phosphorylation, right? Nothing really going on in there. Woohoo! People are texting me. I can't comment. Ian, if you're on your Chromebook, it won't let you comment. You have to be on your phone, according to what I understand. Like, you have to be on your phone to comment or on your own personal device. Other than that, Ian, you can just text me. All right. So when it comes to phosphorylation, ADP in an inorganic phosphate to create ATP, that's phosphorylation, because we're actually going to be utilizing just a phosphate floating around. In this process, you have to end up utilizing a membrane-bound enzyme, ATP synthase. So then we can get more specific about which of the types of phosphorylation. So if you are in photosynthesis, okay, photosynthesis, your only option for phosphorylation is photophosphorylation, okay? The photophosphorylation literally just means that you're going to be doing this with the use of light. Okay, this is the only option in photosynthesis is photophosphorylation. Now, respiration, okay, what you do as a heterotroph, there are two options for phosphorylation. There is substrate phosphorylation, which means that you don't use just an enzyme and add an inorganic phosphate. Okay. In substrate phosphorylation, this doesn't happen because it's not inorganic just sitting around by itself. In substrate phosphorylation, that phosphate, okay, that phosphate, Pi, looks like this. Okay. If it's a substrate phosphorylation, that Pi just isn't floating around by itself like this. It's actually attached to another molecule. So that attachment to some kind of molecule fill in that box, and that could be glucose. As glucose is being broken down, right, in respiration, you're actually helping to make ATP with it because you attached phosphates to glucose. If you're in the Krebs cycle, you also did substrate phosphorylation by attaching these phosphates to pyruvate. Okay, so the phosphates aren't floating around, they're just attached. And then, so substrate phosphorylation is glycolysis and the Krebs cycle. Okay, or we have our last one, which you mentioned, oxidative phosphorylation. And that's only in the ETC. Okay. All right. So glycolysis and Krebs, that's only found in that's the substrate one, but then oxidative, that's only ETC. So this is similar to photophosphorylation, where we're going to use light to get energy to phosphorylize. But down here, we're actually going to use oxidation to phosphorylize ADP. Okay. So the oxidative phosphorylation is draw out your ETC. That's what we acted out today. Let me actually kind of draw my picture a little bit better for you. Okay, we acted this out. When we did our membranes, our membrane proteins, and y'all were fixed proteins versus mobile proteins, okay, and your ATP synthase. In the ETC of cell respiration, this is oxidative phosphorylation. Like this whole picture, I can look at it and say, hey, do you know what's happening in this picture? Oxidative phosphorylation. How do I know that? Because I'm like super smart. Because, I stayed awake in class and I learned. Okay, so in this picture, ETC, oxidative phosphorylation, because you're going to provide energy by taking NADH and FADH2. You're going to provide energy by taking these two molecules and oxidizing them. That's why our two molecules are sitting next to those proteins, passing their markers, oxidized, reduced, oxidized, reduced. And the electrons, that's an E. The electrons now flow, creating hydrogen gradient. Hydrogens get pushed up here. They flow through chemiosmosis, and ATP is made. Okay? And the only reason ATP got made is because of these energy molecules that got oxidized in the first place. So it becomes oxidative phosphorylation. Let me know if I need to explain anything else about that, Alicia. I can't even open it on the Chromie. It says restricted. See, that's so freaking annoying. I emailed them and told them the issue we're having with it. And they're like, we'll see what we can do, but really, I don't know if we can do that. And I'm like, okay, well, thanks. Whoop-de-doop. Sorry. Can you explain the non-cyclic cycle? How it moves back and forth between PS1? Yep. At Michael, yes, T. That was at Michael. Yes. Mm-hmm. All right. Photosynthesis and its ETC. So if you're looking at PS2, cytochrome, that was the only other protein that I needed you to know, cytochrome. PS1, and then ATP synthase. Okay, I'm sorry, these pictures are just kind of, the questions are building up. PS2, cytochrome. PS1, ATP synthase. This is your thylakoid membrane. This is ETC. If you're going from, light exciting an electron and just passing it through and going down, la, 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 there you go. And right here you make NADPH. Ding! This is non-cyclic. In a non-cyclic, we utilize all proteins, we make NADPH, and we make ATP in a non-cyclic situation. This is a healthy plant. This is a healthy environment. Non-cyclic is. Cyclic, however, will happen when there isn't enough water available. So this little lightning strike, right? Remember, or not lightning strike, but the photons as it gets absorbed. PS2, that's where you actually break water, right? But if you don't have enough water available to create this gradient, then you're going to skip all of that. PS2 won't exist if you have to go through cyclic. In a cyclic, you get rid of PS2, so you're not breaking water anymore. What you're going to do is you're going to excite electrons. Those electrons are going to then pass back through the cytochrome and come back around. And they're just going to keep going around and around and around. They get excited in PS1. They get excited because you're still absorbing light energy. Light still comes in, still gets an electron excited, but instead of the electron getting passed down here to the NADPH or NADP reductase, the electron gets passed up here to the cytochrome, gets excited by the cytochrome, and guys, the cytochrome actually has capabilities of making ATP itself. So in a cyclic, you don't get to use PS2 because you don't have enough water. But you're still a growing plant. You still need to survive. So these plants will modify their electron transport chain, just go through the cytochrome, keep going back around so they can get a small amount of ATP. It's not a lot, but it's survival mode at this point in a cyclic situation. Okay. Burial stuff. Okay. Non-cyclic is the ETC. Yes. Cyclic only produces ATP because no NADPH is getting reduced. Yes. Since photosystem 2 isn't being used. Perfect, Jacob. Where in the ETC did we get water as an output? That's your ETC. That's your oxidative phosphorylation, because in photosynthesis, your final electron acceptor is your NADP. But for us, okay, for us at the end of our last protein, oxygen is sitting here. And this oxygen molecule becomes our final electron acceptor. So these electrons travel. They get grabbed up by the oxygen molecules. As the hydrogens flow through, the hydrogens plus the electrons plus the oxygens give us water. You make water at the end of your ETC as an animal in cellular respiration, while a plant would end up making an energy molecule, NADPH. See, it's opposite. Plants made energy. We had to use up that same kind of energy. Okay, other than that, I got it. Okay, the electrons get with the oxygen at the end and make water, Alicia. That's right, Patrick. Butchered your name. My bad. Yeah. Okay. Okay. I meant cyclic. Yeah. Again, Megan, hopefully I just answered that for you. The cyclic, right? Boom, boom, boom. ATP, no NADPH. Just like how cyclic unemployment is unhealthy, i.e. a recession. Yeah, sure. Uh-huh. Yeah. Okay. How does the cytochrome have the ability to make ATP? Cytochrome, it's called a cytochrome complex. It itself is a multitude of proteins within it. And when it takes those electrons back around, it's able to produce the energy to add a phosphate to an ADP. Ding! And just like the ATP synthase, hydrogens, right, provide the energy there. The cytochrome does. It's not as efficient as it's supposed to, as ATP synthase would be. Let's see. We've got a couple of questions. Somebody text me. We have to just turn in the 2 charts. I think that's John, not the review. Yeah, you never turn in a test review. Yeah, Krish. You never turn in test review. Hey, can you explain non-cyclic? I know it is low water, but I don't know how it moves from PS1 and back. Here's the thing, Maggie. So your electrons in the cyclic, electrons have to go somewhere. Like electrons can't be passed to something. So it's like, so in this option, electrons have to be typically moving forward. So they should have gone to NADP reductase, but that has been closed off. And so it has no other, it's like, and it's got to somehow go somewhere else. So that's when it's just going to detour back and say, okay, that way's closed. And it's going to go right back to where it just came from in the cytochrome. And we're just going to keep on going. Oh, it's still closed. And we're just going to keep on going back to the cytochrome until that enzyme becomes active. Okay. All right. Hopefully that answered that. Make sure to like, comment, and subscribe for more biology streams. Oh, heavens. Okay. Let's see what other questions. Okay. How does the cytochrome? Okay. What is PGA, PGAL? PGA and PGAL, those are just your names of your intermediate molecules when you're going from CO2 to sugar or from sugar to CO2. So when we're talking about the cycles, whether it's Krebs or it's Calvin, you're going to you technically can see, or not even Krebs, actually, it happened in glycolysis. I'm sorry. Again, where you see glucose being made or broken, part of its intermediate steps is PGA and PGAL. Darnell was in my class after, during 7th period, and I walked them through the steps too, but I don't require them to know PGA and PGAL, but they're still there. Like, I still want them to see that this doesn't just magically become pyruvate. Like, glucose doesn't just become pyruvate. Bam! There's actually, it becomes other things in the process because we're changing it, right? So I had it and I just told them, I said, guys, this PGA and PGAL, you don't actually have to know their name. You can draw a rubber ducky for all I care. You can tic-tac-toe in that spot. Just know that there is something there, right? It doesn't just, pyruvate. Okay? So if we look at respiration, here, let's look at, so you go from your 6 carbon, okay? You go from your 6 carbon, and we're going to break that. When we break it, we actually end up utilizing ATP in water to break that. Like, both those molecules are used to chop up the 6 carbon glucose. What we end up getting is this. 2 3 carbons with a phosphate attached. This is your PGAL. right? And then we would convert it again. We do some more things, and eventually it becomes PGA. And then eventually it becomes your pyruvate. So PGA and PGAL, again, those are just other names for your intermediate steps to become sugar. So your PGAL is also your G3P. PGAL equals G3P. And if we're looking at the Calvin cycle, right, we take CO2 in, and we spit out a G3P. And it takes two of those to bind and make a sugar. Okay, so that's where you're seeing these names, Kat. Is there any chance we can just go ham on the charts on the back of both worksheets? I feel like that will help understand what everything does. Is there tutoring before school? I mean, if I get there, but Fridays aren't I'll answer your question, Patrick. Fridays aren't a typical tutoring day, so my butt does not rush by any means. If I'm there and you're there, great. Okay, I won't throw you out by any means. Definitely come into the room, use the board, whatever you need to do. What time I get there, it may be 8. Okay, all right. But if I'm there, come on in. Okay, so Patrick, you want to do the charts on the back? Like, what do you mean? Here's the thing. The charts on the back, guys, not everything's going to have something filled into the box. Like, some stuff does get used. Some stuff doesn't get used. Some stuff is a product. Some stuff isn't. Okay, I can definitely pull it up, Patrick, and just kind of look at it real quick while y'all are asking your questions. Okay, you do not have to know specific numbers. You don't have to know 12 ATP, 9 ATP, 9 NADH. You don't have to know those things, so don't worry about that. Don't get super stressed. Oh, y'all. No, I'm exhausted. Like, I don't know what it is with Halloween that just exhausts me. I'm not sure. Okay, let me pull this up real quick. All right, so specifically, what do you think are some of your questions? Like, can you generalize and look at the photosynthesis equation, right? Can you look at that and tell me where you can see those reactants and where you can see those products, right? So like if you're looking at respiration, okay, if you're looking at respiration, can you tell me where you would see glucose and where you would see oxygen? Like, can you tell me different pieces of that? Don't get super stressed, Miss Good, as Patrick chugs down second Red Bull. Patrick, I'm sorry. Please, I don't like Red Bulls. They make y'all's heart race too much. Okay. All right. So can you tell me where glucose and O2 are seen as reactants? Keep that in mind. As a reactant, meaning you're going to be utilizing them. So glucose, that's where you're going to see them in glycolysis, right? If you can recognize that, you're going to see it in the cytoplasm, right? And like, I mean, that's kind of as much as I would really want y'all to know in this little step. Like, can you at least do that? Okay, that glucose is glycolysis, and you see it happen in the cytoplasm. While oxygen, that's our final electron acceptor. Okay, oxygen is your final electron acceptor in respiration, and you're going to see that in the ETC in the cristae. Okay, see that as a final electron acceptor in the ETC in the cristae. Again, this is as reactants. You know I'm talking about them as reactants because they're before the arrow. And, like that's how you know it's a reactant. And then can you do the same thing for the products? Tell me where you see CO2 and tell me where you see water. So because we release that CO2, the CO2 you're going to see as a product of pyruvate, oxidation, and the Krebs cycle. That's the purpose of both of those, is to release CO2. And that's all going to happen in the matrix. Okay, you release CO2 in pyruvate oxidation, sorry, that says pyruvate, and in the Krebs cycle inside the matrix of the mitochondria. H2O, that's where you're going to see it produced, right? Water is being produced because of the oxygen acting as an electron acceptor in the ETC. Okay, that's all it is. Water ends up being in the ETC and the cristae. Right? And then do the same thing for photosynthesis, right? And just kind of flip that. Where do you see CO2 and water being used as reactants? CO2, you're going to see it used in the Calvin cycle, in the stroma. While water, you're going to see it broken at photosystem 2 in the light-dependent reaction. All right, light-dependent reaction, photosystem 2, not photosystem 1. You only see water broken in photosystem 2. And so if this is photosynthesis, right, you see CO2 in the Calvin cycle being used inside the stroma. You see water in the PS2 in the light-dependent reaction in the thylakoid membrane. And then glucose in a plant, you end up seeing made in the Calvin cycle, in the stroma, also known as pGal or G3P. Okay, Calvin cycle, stroma, made in pGal or G3P. And then oxygen getting released, it becomes a waste, right? It's getting released in the thylakoid membrane as a waste during the light-dependent reaction in PS2. Okay, water gets, oxygen's a waste to a plant. They don't get to hang on to it. Thylakoid membrane in PS2. If you can kind of do this, this is definitely like the first step of just being able to organize that entire chart. I will get to more questions about the chart, but let me go ahead and answer some questions I see on here. Okay. Ew, Red Bull and coffee? Ew. What's, no, people. Glucose is in glycolysis to make pyruvate. Yes. And some ATP. Yes. You make only 2 ATP, or you make 4, but you only net 2. O2 is as an acceptor of electrons, right? Yes, oxygen's the final electron acceptor there at the end in the cell respiration. PS1 doesn't make ATP. No. Yeah. PS1, all PS1 is doing is absorbing light energy. It's going to absorb to pass the excited electrons one more time, this time to the NADP reductase enzyme to make your NADPH. So he's just passing electrons. He doesn't break water. He doesn't make ATP. Only the cytochrome can make some, and the ATP synthase can. Or does it because of non-cyclic? Again, PS1 can't make the ATP. That's the cytochrome complex that does. If you mix Red Bull and coffee, it's a guaranteed 100 on the test. People, please really don't drink that. That's not good. That's a lot of caffeine. You're too young. How does a plant make ATP if no light is available? Okay, if no light's available, so the questions that I end up wanting you to be able to answer on something like that is kind of look at a couple of different aspects. You can come at it, a couple of different ways. If you are a plant that has taken on, what has just happened? As a plant, if you are a seed, meaning you're under the ground, you can still make ATP, but you're not actually going through photosynthesis. You're going to be going through respiration. Plants have the capability of doing respiration just like we do because they have mitochondria as well. A plant that is like maybe in the bottom of the ocean that doesn't have access to sunlight isn't actually going to be able to go through photosynthesis, but it's going to be able to go through chemosynthesis. Those things that like the hydrogen, sulfur bacteria and stuff that live in the bottom of the ocean thermal vents, those organisms make ATP. They are autotrophs. They just use chemical energy instead of through like a sulfur gas. They use that instead of light energy. Okay. Let me see if I... Seeing if I have anything else. Let me look at this. Come on. Okay, Kat, did that answer your question for you? Boo, 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 boo. Okay. All right. So your plants end up ultimately just having to utilize either stored energy and go through the mitochondria or use chemosynthesis. So that's the 2 chart worksheets due tomorrow. Yes, those were your worksheets for the chapter. Those are your worksheets are always due on test day. Okay. Yes, we will need to know. Will we need to know about CAM plants? All you have to know. So photosynthesis is... It's like you just thought when you took AP Bio that you learned photosynthesis. And then I come around and say, nope, there's actually 3 kinds. And you're like, I want to cry. It's okay, guys. What we've been talking about. So photosynthesis, there are 3 kinds. We call them C3, C4, CAM. When you learn photosynthesis, you're learning this guy, C3. So when we talk thylakoids, when we talk mesophyll cells, when we talk rubisco, RUBP, all that stuff, right? Cyclic, non-cyclic, that's this guy, C3. C3 is your big kahuna. It's the easiest, it's the most abundant, and it's the most energy efficient photosynthesis. So that's what plants want to be, is C3. But look at that. Some plants haven't been able to be that, okay? So here's why they're different. Your CAM plants have had to adapt to arid climates. And Dylan, if you're on here, I misspoke earlier this afternoon. So if you're listening to me now, right? Okay. So your CAM plants have had to adapt to arid climates. So these are going to be your desert cacti and agave plants, pineapple plants, those kind of things. Things that have had to adapt to arid climates, so very dry desert kind of like climates. They aren't necessarily going to be the most photosynthetic, if that kind of makes sense sometimes. They won't be the most photosynthetic organisms. They're going to have a high water concentration. Think about a desert plant. What do they have to do? Hang on to their water. So a CAM plant simply is a special kind of photosynthesis because what's had to happen is they have to hang on to their water and they go through a different process of carbon fixation. They close their stomata during the day. Okay? They close stomata during the day. And if you think about it, the stomata are for gas exchange, but water can leak out too. So how do they avoid this? They close them during the day because they're in a hot, arid environment. So it only makes sense to close it. So all I need you to know about a CAM plant is it's a different type of photosynthesis. You would see it in arid plants. C4, and so what we say is they are temporally separated. A temporal separation literally means time. C4 is spatial. Spatial separation in a C4 means that they use the bundle sheath. Okay, so 3 kinds of photosynthesis, most common, uses a separating space, so they use a different part of the cells. Can plant is time separated, closes stomata at the day. Other than that, like just knowing that there's a spatial and a temporal separation of photosynthesis and that 3 kinds exist, that's all I would need you to do. Okay, nothing more than that, Keaton. Do we need to know any examples of mobile proteins? No. Plastoquinone and viridoxin and no. You already have plenty to memorize. You don't need to know those names. Okay. Just that they exist. Yep. Good job, Patrick. Is PQ1? Yep. Plastoquinone. Don't need to know it. And they pass off electrons to the fixed proteins. Yep. That's all they're doing. They're the ETC. Your proteins in ETC for you as a heterotroph are called fixed immobile. Like that's all. And they just pass electrons. Then the ETC in photosynthesis. Yes, I wanted you to know those names. PS2, PS1. PS2, PS1. That was it. Yeah, that's what I meant. Exists. What? Rubisco is short for the really long thing used in carbon fixation. Yes, it is. Well, okay. Rubisco is the enzyme that helps to grab a CO2. The CO2, right? CO2 comes into the cell. It's like, whoa, where am I? And it's in this big open space of the stroma. And Rubisco, as a very sweet enzyme, wants to help out CO2. And it says, oh, let me help you. You know what? I know someone you need to meet. So CO2 grabs Rubisco and says, hey, I want you to meet my friend, RUBP, 5-carbon molecule. And he goes, ding. And Rubisco helps to bind a single carbon to 5 carbons, making, well, the precursor to glucose, right? So Rubisco helps to guide, kind of like acetyl-CoA. The CoA helped to guide the 2 carbons to the oxaloacetate, the 4 carbons. You see Krebs and Calvin cycle, you've both got somebody escorting some kind of carbon into the cycle, and they have to be joined with a carbon that already exists there. So let me kind of photosynthesis versus cell respiration. Calvin cycle versus the Krebs. Are you, woo. I can't write today. Okay. Okay. All right. So in the Calvin, so I'm just I compared. So Calvin cycle versus Krebs. Oxaloacetate, I just wrote oxalo. Oxaloacetate, CoA, live there. Like they just live there. They're just always like roaming around. La, 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 right? What happens is all of a sudden, whoa, who just came in to our space? Whoop. Nobody noticed that I completely flipped those titles, did you? No, we're just going to. Give me just a second. Okay. All right. Let's do this again. So I made this little chart that's actually right now. So in photosynthesis, you've got this RUBP and Rubisco just like floating around, la, 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 living their life. Well, then all of a sudden, Oh my gosh, who just came into our space? CO2, who are you? Rubisco, get into action. Rubisco comes over and says, oh, hey, who are you? I think you need help. Let me take you somewhere. And so CO2 comes over by the help of Rubisco and gets added to RUBP. RUBP grabs him and is like, I'll take you, buddy. I got you. And so this 5-carbon molecule, 1, 2, 3, 4, 5, now gets a 1-carbon molecule attached. You've now made 6 carbons. And so they're like, wow, what can we do with you? Haha, we're going to chop you off and make sugar out of you. That's ultimately what it's going to do. And then what happens is RUBP and Rubisco just go back to living their life walking around until another CO2 molecule comes in and they get thrown into the cycle, because the Calvin cycle does just that. Takes in CO2, uses RUBP, an enzyme helps bind them, and the Calvin cycle's job is to make sugar and use energy. Pretty much the same thing happens in the cell respiration, the Krebs cycle. They're just living their life. Oxaloacetate and CoA, they're just living their life in the matrix. La, 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 la. I just love it in here. And then all of a sudden, what has entered our space? Pyruvate. Who are you? Why are you here? Where did you come from? 50 million questions. Answer them now. So CoA is like, hey, buddy, I know where I can take you. Don't worry. I'm going to help you. CoA takes the pyruvate first, oxidizes it, and says, ha, I'm going to chop you up I'm going to go ahead and take you real quick, and we're going to convert you from 3 carbons. Yep, no longer 3 carbons. Yep, we're going to go ahead and chop off one of you, and you're left as 2 carbons, and I'm going to guide you, CoA says. I'm going to take you to a friend of mine. I know this has turned really dark really fast. I'm sorry. And it takes it over to the 4-carbon molecule, oxaloacetate. Where am I? Takes it to the 4-carbon molecule. CoA says, hey, meet my friend. Here you go. And now all of a sudden you make a 6-carbon molecule that's going to release CO2 and make energy. Okay. So both have these molecules that always live there. Rubisco, RUBP, oxaloacetate, CoA. Those always live there. They're just waiting to do their job, which is to take in the molecule, add it, convert it to whatever they're supposed to, and release the RUBP and oxaloacetate to regenerate again. We only really name the stages of the cycle in photosynthesis as fixation, reduction, regeneration, right? Your Krebs cycle doesn't really have those names. All right, give me just a second. There's a few questions that came in. Okay. Yay, that's what I meant by okay, Rubisco, thanks. Yay, Ian, you got it right. Easy 100, all right. Can you show NADH being produced in glycolysis? Yes. Can you? No, I'm just kidding. I'm like, this kid, we do not have time for your sass. All right, let me go ahead and get that going. Where is the chloroplast gradient developed? Is that just the membrane? Yeah, that's just on the inside of the thylakoid membrane. Your proton gradient, that's all that's going on. Chloroplast gradient's the buildup. Yes, good job, Megan. Is it going through the synthase yet? I think gradient, is it going across? Yes, so that's being pushed. You guys that were being forced up, you were creating a gradient, okay? And then you came through the synthase, because when you came through the synthase, that was just diffusion. You went against your gradient when the electrons passed and you got pushed. Okay, you got activated, you just kind of got pushed. I don't know if going through the synthase is a part of it, but the gradient is them all being smushed. Yes, just them being smushed. It's what we did when we piled up against the wall. Yes, good job. Good job. Okay, NADH. Megan, let me get that for you through the glycolysis. So Glycolysis. Take your 6-carbon molecule. So again, we're going to chop it. To chop it, we're going to use ATP. We're going to use water. And what that's going to do is pull them apart, but ATP gives up a phosphate. So you get this guy, right? So far we've had to use 2 ATP, and it gave up phosphates. You're like, interesting. Huh, why did it do that? Well, here you go. Okay. All right. And then ultimately, another enzyme. Okay. An enzyme. Don't worry about this, but an enzyme is going to come up and add. Well, let me go ahead and do this first. It's going to do it in a second. These phosphates don't stay there. Those phosphates are ultimately going to fall off with the use of enzymes. And when they fall off, what they actually end up leaving, okay, an enzyme helps to take off those phosphates that just got added. But in addition to taking off those phosphates, they also end up releasing a hydrogen molecule, unfortunately. So all of a sudden, that free hydrogen has to go to an NAD plus molecule, and that ends up becoming NADH. Okay, so you end up making 1 NADH molecule total, and I could get into the specifics of why it actually only comes off of 1. It has to do with the functional groups that are on here. This one of your carbons ends up being just fine. He's perfectly happy. The other one isn't. Hydrogen has to come off of one of them. So you make 1 NADH, and then we'll end up adding phosphates through an enzyme, rip them off, and you end up making 4 ATP at the end. Okay, is what we did. Okay, yeah. Could you explain gluconeogenesis and how it relates to anabolic pathways? Check this. All gluconeogenesis is how we will still go through cell respiration, like you still go through glycolysis, pyruvate oxidation, Krebs cycle, and the ETC. You go through all those steps no matter what. But that's it's going to go through all of those if you're going to use glucose. Oh, looky there. Sometimes you don't take in any glucose, especially people who are like being extremely health conscious. They may be trying to minimize how much glucose they take in. So they may be eating just a whole lot of fat. They may be eating a whole lot of protein. So what happens? You still survive somehow. You still go through cell respiration, but wait. Glycolysis, pyruvate. Glycolysis is the breakdown of sugar. Well, if you don't have sugar, you can't go through glycolysis. So in gluconeogenesis, what happens is proteins get broken and they might enter pyruvate oxidation. They may enter directly into the Krebs cycle. They skip glycolysis. They skip pyruvate oxidation. Whatever molecules you take in, whatever food you consume, you're going to go through cell respiration one way or another. You just might get to enter it at a different step. Okay, you're still able to make ATP from the cream cheese you ate with 0 sugar. You still got to eat using that turkey stick that you ate. Okay, you're always going to make ATP. You just might enter it at a different stage. All right, the 4 ATP of glycolysis, is that where that has a net gain of 2? Yes, because you had to use 2 to break it in the beginning, so you had to pay yourself back. And that's really, guys, that's really the only ATP number that I'd really like ask you to memorize. What is ADP and PI? PI is the inorganic phosphate, a product of PS1, 2, or Calvin. That's going to be a product of the Calvin cycle because the Calvin cycle uses ATP. So when it uses ATP, it releases ADP and a phosphate. Okay, it's got to take that phosphate off to get energy out of it. Okay, your questions are kind of slowing down. I guess y'all ran out of Red Bull. See what happens when you drink Red Bull? You crash. No. Yes, y'all do. Okay. There's always no. The night is young. Oh my God. I think I was old even when I was a teenager. Oy. I had no problem coming home and just going to sleep and just like chilling. Put on some this is gonna date me. Oh yeah, this is gonna date me. And y'all probably have no idea what I'm talking about. Old TV show back in the day. It was called oh gosh, Third Rock from the Sun. It was an old comedy. Like these aliens living among us as real people. It was stupid and funny. I would come home and watch that. The basic function of fermentation to recycle NADH. Okay, to recycle the NADH because glycolysis makes ATP and it makes NADH, but it can't, but it glycolysis' ultimate job is to make your ATP at the end, right? So, so in glycolysis, its ultimate purpose is to make 4 ATP. Okay, so glycolysis, its ultimate purpose is to have used 2 ATP, but created one NADH and create 4 ATP. This is the main goal. Get down here. But to do that, you have to go through these two first. So fermentation's job, sorry, I'm trying to get more comfortable. Fermentation's job is to recycle this as NAD so you can get to here. Because in fermentation, right, we only, my husband's watching something weird. In fermentation, there's only 2 pathways we talked about. Alcoholic. In alcoholic, it released CO2 released, and made alcohol. Okay. Alcoholic released CO2 and made alcohol. This is only if you're bacteria or fungus. But for you, you actually go through lactic acid fermentation. And the purpose of lactic acid is to make, well, lactic acid. Okay. But here's what happens. In fermentation, you're actually taking a 3-carbon molecule. You're taking pyruvate. You're going to convert it into a 3-carbon molecule called lactic acid. I know they look the same, but they're not. There's conformational stuff, extra oxygen here, extra hydrogen here, that kind of thing. So you went from this 3-carbon to this 3-carbon fermentation. But to do that, you had to use NADH. You had to use an NADH molecule, which then meant you got to release NAD, right? So now that NAD can flow back to glycolysis to get made again. So it can get made so you can get here to the 4 ATP. Fermentation helps to recycle NAD so you can make more ATP. That's why you failed your test in college. Shouldn't have slept. Oh yeah, I slept all the time. Oh yeah. Actually, I very much remember my first cell biology test. I want to say it was cell biology, maybe biochem. I don't know. I stayed up studying all night, literally all night. I didn't go to bed. It was my test. My class was 7 a.m. I stayed up the entire night. I just kept studying. Around 5:30, 5:30, I finally took a shower and stuff. And I went to school and I was like, yes, I stayed up all night. I stayed up all night. I made a 52. No. You're supposed to go to bed. It's actually scientifically proven. You're supposed to get 2 cycles of REM sleep for information to sink in. So you should have started studying on Wednesday. All right. How's glycolysis a metabolic pathway? That's how I am in your class. Sleepy. Yeah. Okay. Glycolysis metabolic pathway because it's part of yours. It's part of the whole process of cellular respiration. Like we don't. This is what's kind of annoying. When you see the title for cell respiration in a book, they immediately start talking about glycolysis. Technically, glycolysis isn't part of cell respiration. What? Miss Good, don't do this to us. Slow down. Don't worry. I'm not messing with how you have it figured out. But because glycolysis is anaerobic, meaning it doesn't use the oxygen because it's in the cytoplasm, it's its own separate beast, but it's still part of the metabolic pathway. We still talk about it, obviously, because that's where it all begins, guys. You can't get pyruvate to go into the Krebs unless you break down some sugar in glycolysis in almost all cases. So it has to be a metabolic pathway because it's how we're going to produce energy. Even glycolysis gets us to ATP. Gross. Anyways. Okay. What are the overall functions of PS2, PS1, and Calvin? Am I just answering your chart for you, Patrick? Is that basically what just happened? Overall functions, PS2, what do you think? It's the very beginning of the ETC. It's going to absorb energy, but its job is to... Waiting for you to respond. No. It's just... Exactly, it is! Yay! It's just where PS2 breaks the water through photolysis. You're right. Perfect. Okay? That's exactly right. Okay? PS1 helps to pass electrons to the NADP reductase, or NADPH, in non-cyclic. NADPH in non-cyclic. It doesn't go to it if it's cyclic. Okay, so PS1, it's just NADPH is helping to create that. And then the Calvin cycle, it's going to use the energy you created and help make sugar. Okay. Yep. Ding, ding, ding. Patrick, be smart. Yay. The Red Bull hasn't killed the brain cells yet. Yay. Okay. They'll bring hydrogen ions over when they exchange electrons in ETC and make the gradient. Correct, Ian. We can not ATP, ADP, stupid autocorrect. Oh, makes ADP. Yes. It makes ADP, not ATP. Yes. Good job. Got such a long night ahead of me. Oh, wait, so do you. Guys, do not stay up stupid late. We're already up late doing this. This should be enough, okay? So NADH and NAD are used or produced in every part of respiration. Yes. Glycolysis, yes. Pyruvate oxidation, yes. Krebs, yes. ETC, yes. They are. They're either used or produced because they're oxidized form or the reduced form. Correct. I'll be in by 2, asleep by 4. Oh. No, don't do that. Don't do that. I can have pixie sticks ready for y'all in the morning. Y'all can chug down some pixie sticks and get super sugar rush. Yes, right before the test. Okay. What else can I answer? Nope, we're good. Done what we can. Kind of reviewed it, answered what we could. Yeah, okay. Take one of Mr. Bean's coffee bean jugs. Coffee bean jugs. What is that? I don't even know what that is. He has a jug full of coffee beans that he like chews on? Like he just chews on coffee beans? He literally just eats beans? Really? Oh, wow. No, he doesn't. He does not eat raw coffee beans. Comes in the name. He does not eat raw. Now, some people actually do eat chocolate-dipped coffee beans, and they're supposed to be really good. He is not eating the coffee beans. Oh my gosh, that's just funny. He does not use the grounds like dip. All right, products of the citric acid cycle, the Krebs cycle. It's releasing CO2. That's the big one. So, thanks, Krebs cycle. You just released my CO2 for me. All right. Produces CO2 half. Oh, heavens to Betsy. That's just funny. You know that, T. So the citric acid releases CO2, and then it helps to make energy. FADH2, NADH, and ATP. And the NADH and FADH2, those molecules came over to the fixed proteins and started becoming oxidized, passing their electrons. Okay, those came directly from the Krebs cycle. What else are we doing? Where do the problems come from? Okay, where do the protons come from in respiration? From the oxidation of NADH and FADH2. When they got oxidized, they lost their electrons and their hydrogen. Remember, always follow electrons with hydrogens. So those hydrogens got ripped off, they got pushed, they come through, and they're also being released because water, when it gets made, it's going to be used up somewhere in some kind of chemical reaction and release its hydrogens. Role of redox acids in photosynthesis. Your redox stuff, so whenever you're looking at oxidized, reduced, in the Calvin cycle, you're going to end up using that NADPH, and you've got to reduce and oxidize it to help make the glucose, ultimately. Also, your ETC, the NADP reductase enzyme in the membrane of the ETC. How the photosystem 1 passes electrons to that enzyme, it helps reduce NADP and add an H to it. Okay, so you still see redox reactions all in the ETC and the Calvin cycle. You just see them more prominently driving the ETC inside of you during respiration. Yes, NADH and FADH2. Good job, Patrick. Good job. All right. Boo, 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 boo. Are we winding down? We feeling like we're done? I don't know if there's anything else we can do. Anything else? Yeah, well, we're gonna have to wrap it up here pretty soon, Ian. Sorry. I can't stay on here all night. I mean, I could, and, I could just, leave the camera going, and y'all could awkwardly watch me, sleep and toss and turn. I mean, that's just creepy, but yeah, we're going to have to end this at some point. Okay, let's see, what were the questions? As oxygen carriers, are oxidized, are electrons released? Let's correct that just slightly. They're not oxygen carriers, they're energy carriers. So yes, when NADH and FADH2 are oxidized, they released electrons because the hydrogens were being held on by electrons. So when you pull off a hydrogen, you also pulled off 2 electrons as well. That's why y'all were passing the 2 markers to each other. Those 2 markers represented the electrons. What happens to Rubisco when oxygen concentration is much higher than the CO2 concentration? Remember Rubisco? Its dirty little secret, right? Rubisco's dirty little secret. It loves oxygen. So if there's more oxygen available than CO2, Rubisco's going to grab oxygen instead. That's not good. That's called photorespiration, aka equivalent to cell death. Not good for a plant. Because if they don't grab CO2, they don't make glucose. They don't make glucose, they don't build cell walls. They don't grow. Okay? Jake Poole ASMR. What? I don't even know what that means. Okay. Wait as electron carriers are oxidized. Wait. Yeah, okay, good. Does fermentation occur where glycolysis would occur? Yes, both in the cytoplasm. Correct. Good job. Seeing if there's any questions that are like that we haven't really answered that I could point out for you. Let me see. I think we've gone through like everything. Know the 3 parts of the Calvin cycle: carbon fixation, reduction, regeneration. Okay. I think we kind of gone through everything. I looked at it. My tummy hurts. I'm sorry. Go tell your mommy. Where does Rubisco take O2 when it grabs it instead of CO2? It's extra points for the 12 survivors tonight. It's the Rubisco. Let me kind of walk through this real quick in my mind. When the Rubisco grabs it, it's still going, like it still tries to add it to RUBP, like it's still trying to go through the Calvin cycle, but it can't, it doesn't actually end up falling off as a G3P. Okay, so it grabbed the oxygen, but it can't actually create a carbon skeleton for glucose out of it. So it grabs it, and eventually another enzyme is just going to remove it and release it as a waste product. But it tries. Rubisco desperately tries to put it into some kind of form that's usable, but it can't because it needs the carbon to make glucose. Technically, it doesn't matter, Ian. If oxygen goes into ligma, what happens? I don't know what that means, ligma. I don't know what that is. It doesn't really matter, but that's okay. The carbon skeleton is G3P. Yes, Ian, you're so smart. You got it going. That's awesome. Why is that funny, Patrick? Why is that hilarious? You don't think that Ian's got this? I think he does. He's got it. All right. I really do think y'all have gotten as much as y'all can. Y'all studied like plenty because y'all were pretty much, ladies and gentlemen, we got them. What? Possible dates? photosynthetic products. What does that mean? Where in heaven's name does it say? Oh, fates. I was like, when did I talk about dates of something? The fates. Yeah, all that's meaning is like, so you produce oxygen, and so oxygen leaves, right? It's gone. It's a waste product. But then you produce glucose, and glucose goes into the cell walls as structure. That's how plants grow. Dead plants, fates. You're good. No, you're good. Okay. I think y'all really have gotten everything out of me y'all possibly can. Other than that, y'all just need to maybe review a few things or draw out, but I really wouldn't stay up any longer studying, guys. If you were able to follow the conversation, if you were able to answer each other's questions, then I think you know everything. Like, said photosynthetic was bad. Photorespiration is bad. Photosynthetic as in photosynthesis is good. That's how plants live. They've got to go through photosynthesis. But photorespiration, where Rubisco grabs O2, that's bad because then it's not it's not making glucose like it's supposed to. Okay, Patrick? Good. All right. See, we're screwed. Why are you screwed? What just happened, Patrick? I don't understand. Photorespiration is no CO2 for Rubisco, correct? It grabs O2 instead. So think about it. Photorespiration. It's a plant like that's breathing. It's grabbing oxygen when it should have been releasing it out. It should have been getting rid of it as a waste product. But when a plant accidentally holds on to it, we call that photorespiration because it's breathing in the oxygen instead of the CO2. Mañana. I know mañana. Okay, mañana. All right, talk to y'all later. I'll post this. Bye.